Daniel?
Just a minute. Okay, we are live.
Okay, so thank you very much, everybody, for coming to the seminar, the webinar today that is going to be about the high throughput phenotyping. And this is the 13th class of our course from gene to field, or from gene to trait. And today we have a great pleasure to receive Daniel Schachtman from University of Nebraska-Lincoln. Daniel is the director of the Biotechnology Center, and his research interest is in the interaction of microbes, soil microbes with plants. And he is also managing a big team involved in many aspects of plant biotechnology, using different crops as a model. And today, he organized a set of talks from the research scientists and PhD students from his lab. And Daniel, thank you very much for agreeing and in collaborating with us in our course. And I will ask you to start with the presentation and introduce the three speakers from your center. Thank you very much.
Thank you, Paulo, and thanks for inviting us to speak today in this class. We're very excited to reach students in Brazil. We have many Brazilian students here at UNL, and we are always welcoming more students. So today we'll focus on high throughput phenotyping, and these are the instructors. At our university, we're extremely fortunate to have a very collaborative team of agricultural engineers, or they call the department that they're from biological systems engineering. And so the first speaker will be Yufeng Ge. Yufeng is an associate professor in advanced sensing systems engineer. He's also an Eberhard Professor of Agriculture, and that's recent, as well as his appointment to be the Director of Plant Phenomics here at the University of Nebraska. He did his PhD at Texas A&M and then worked there for a while. After finishing his PhD, he did his MS and bachelor's degree at Nanjing Forestry University in China. The second speaker, also in the Biological Systems Engineering Department, is Yeyin Shi. Yeyin is an assistant professor, in her title or her area is Agricultural Information System Engineer, but also we're extremely lucky because she's our very collaborative drone expert at the University of Nebraska. So she works with a lot of the plant scientists on campus, including myself, as does Yufeng. So Yeyin did her PhD and her master's and PhD in biosystems and agricultural engineering at Oklahoma State, and she did her bachelor's degree in mechanical engineering at Nanjing Forestry University also. So those are the first two speakers. And then the next speaker will be a PhD student from my lab, Ning Chai. She did her bachelor's degree partially here in biochemistry with a minor in business. And then she also did part of her undergrad degree in Malaysia. And she came to the US via this American degree transfer program in biotechnology. And I was lucky enough for her to end up coming to my lab to do research and to spend about 5 years, which she's ending now. As Paulo said, I'm the director of the Center for Biotechnology. I'm also the George Holmes Professor of Agronomy, a George Holmes Professor of Agronomy and Horticulture. And I did my PhD a long time ago at Australian University in CSIRO in Australia. I'm an American, but I went over there to study. And then I did my master's in at UC Davis. And I actually have a bachelor's degree, which is in agricultural economics, also from UC Davis. So phenotyping has really gained a lot of popularity in the last 5 to 7 years. Basically, when you talk about the phenome, it's all the measurable physical and chemical characteristics of a plant and animal. And so what we're often looking for are 2 main outcomes for phenotyping or phenomics. The one major outcome that we're focused on is knowledge for both plant scientists and breeders. So a lot of people, there are several faculty members that are using these methods to map genes underlying particular traits. So it's a very efficient way to phenotype either in the field or in the greenhouse. Myself and some others have also used these methods to gain a better grasp on how plants function, so the physiology of the plant. And I know something that Dr. Arruda is interested in, and also some people here, is using it for studies on how transgenes change the physiology of the plant. And then kind of thinking towards the more applied areas, you can see these tools could be eventually used to develop algorithms and tools for digital agriculture or precision farming. So you can imagine, like in this picture, this future farm, everything is pretty much run by robotics. You have drones that are analyzing the disease pressures in your field, maybe the need for fertilizers in the field. And so really, I think this is where agriculture is going. And in addition to our basic studies, we also hope to contribute there to future farms. Nebraska is the 3rd kind of largest, in economic terms, state for agriculture in the United States. So we're really an important, our studies are really important for agriculture in the US, in the world, but also primarily in Nebraska. And so, we have this vision for this phenotyping at the University of Nebraska. So what we want to do is move from greenhouse studies to field studies. And you'll hear about this PhenoCart, and you'll also hear about this spider cam system that we have. And then that can then be applied to digital agriculture, plant breeding, predicting genome to phenome, and then analyzing changes in transgenic plants. So today, the topics that we'll focus on is, we'll talk a little bit about the facilities at the University of Nebraska for phenotyping, and and also some examples of how those facilities have been used, including talking about the cameras and sensors. And then, we realize that a lot of you don't have access to these expensive facilities that we're lucky enough to have here. And so, and they're also very expensive to use. So some of us have developed, probably with the help of Yufeng mainly, there'll be a few slides on 2 more manual, low-cost systems for the field and the greenhouse. And then, our engineers here, in this case, Yufeng, has really helped us develop these systems. And then finally, we'll round out with a longer talk about drones and the sensors that are used in drones. So there may be a few parts that are a little bit repetitive, but I think it'll help just reinforce some of the major points that we're trying to bring out. And so with that, I'll switch over to Professor Yufeng Ge, who will lead off the talks today, the teaching.
All right, so can you all hear me okay?
Yes.
Yes.
Okay, I'm sharing my screen and then I'm gonna play. The screen is okay also?
Yes.
Okay. Yeah, so good afternoon, everyone. And like Daniel pointed out, so my name is Yufeng Ge, and I am here at University of Nebraska-Lincoln. And a lot of my research is to work with engineers and plant scientists to develop new plant phenotyping tools and algorithms and help with the plant science discovery as well as things related to precision agriculture and digital agriculture. So Daniel mentioned about that we are lucky that we have this wonderful phenomics core facility here at Nebraska, right? And mainly we got the three systems, right? So on the left that you see, it's a chamber-based imaging system, and that is small and mainly for the model plants such as Arabidopsis. And then in the middle, you can see, and this is an imaging greenhouse, and you see all these corn plants sitting in the pots, and the pots moving around the plants, and then send them one by one to an imaging chamber and take images of different types. And then you can see an example here that is just coming out of that fluorescing imaging chamber. And this is really designed for agricultural plants, right? So bigger than Arabidopsis, corn, soybean, wheat, sorghum is our main crop here at Nebraska. And then the one on the right, as you can see, that is what we call the new spider cam field facility. And this is built on 1 acre field. And then you can see you can't see it from this perspective, but there is a centerpiece that is a sensor platform that we have 4 or 5 different camera types that we carry the information. And with all these, right, so we can do basically 2 things, right? So one is the forward phenomics, and we translate novel genes from the model crop to, from the model plants to agricultural plants. And then we also do reverse phenomics, that's the novel traits discovered in the field, and then dissect them to the genetic basis and functions. So I'm gonna play 2 videos for you that you can see. Sorry about the technology. I'm trying to play this video.
This is our limited tech plant phenotyping facility attached to a 6,400 square foot greenhouse with 672 plant capacity. The imaging system can image plants up to 2.5 meters on a daily basis. Robotics of the system move the plants around, water the plants, and also facilitate the imaging by lifting and rotating the plants. This completely automates the process. So unless there's any problems, we can grow the plants from seed to adults without any interference whatsoever. Currently, these plants are undergoing an imaging job, and the system brings the plants in through the imaging chambers, first through the infrared chamber, then the fluorescent chamber, then the visible, then the near infrared and hyperspectral chamber, and then they come to the watering station. It picks up the carrier, the plant, and the soil and weighs everything as a package and compares that to a target weight in the database. It'll replenish exactly the amount of water it used in the previous day, and we can adjust that target weight higher as the plant gets bigger. We can lower that target weight and very easily drought stress the plants just by changing a value in the database. Everything is computer controlled and monitored by computers, so we can keep track of exactly what the conditions were in the greenhouse over the course of the entire experiment.
All right, so that is a nice video that introduced so that you have an overview of our laminar tech phenotyping greenhouse system. And then I have another one here that introduced the spider cam field phenotyping system.
With this system, we can see what people cannot see by their naked eyes. We can do very accurate measurement and very fast.
The system requires 4 different poles. The tensioning on the cables has to be adjusted based on where it's at in the field. And so it shortens and lengthens each one of those cables specifically to get to a specific location.
We can do any crop we want because it can raise from ground level to 9 meters above the ground.
We can control within about plus or minus a centimeter of placing that camera in the same position each time. So therefore it allows us to get images that are identical from one time to the next in order to make comparisons.
We have minimum shadow on it, which is very important.
With the large payloads, we can do a number of different instruments at the same time as opposed to a drone where you're limited by the payload capacity of the drone. This allows us to do different camera systems and imaging at the same time on the same plants.
We have the very advanced subsurface irrigation system.
We have 130 different individually controlled irrigation zones out in that field.
And we can control independently for all of them. This is the first system in the United States. It will give us a lot of innovative way to develop our modern phenotyping pipeline.
Yeah, that is our field phenotyping system. And I just realized that in Brazil, you are probably using metric system, right? So 1 acre is about 0.4 hectares. So if you are not familiar with the English. So I figured that I throw in some examples about what we can do with those phenotyping facilities. So first to go with the greenhouse system. So here you can see on the left that is a sorghum plants, right? So and then it's a single plant image and it's a RGB image. And then what we do usually is we do the image segmentation, and from there we can acquire a bunch of digital images, right? Digital phenotypes, so including things like a total projected area and height, width, solidity, and essentially a lot of the digital phenotype is better than the manual phenotype because there's a lot of things that you cannot measure easily with a ruler or with a scale, and make it even better is that you can take a series of RGB images, and then that essentially enables the dynamic tracking of the growth, biomass accumulation, and stress responses. So I show you essentially a time sequence image here, and you can see day by day the crop is growing, and then on the left here it's a normal plant, and on the right here that plant is pretty water stressed. And you can see at the later growth stage, about 17 days after planting, their growth trajectory starts to diverge, right? So the one on the left becomes larger and larger. And then we can also track the water use, because of the instrumentation on the system. You can see how on a daily basis, the water use in terms of gram, the normal plants versus the water stressed plants. So that's the RGB image. And we also have a thermal infrared imaging system with the greenhouse that quantifies the leaf temperature of the plants, right, due to water stress. So I gave you 2 examples here. So on the left here, that is a plant with a normal water application rate. And then on the left here is the one subject to water stress. Now you can already see, right, from their size that the normal water plants is a little bit larger with a larger leaf area than the stressed ones. But more importantly, if we look at the temperature information of the plants captured by this thermal IR camera, and you can see that in general, the well-watered plants are cooler, right, than the stressed plants. So that gives us quite useful information and as a rapid and non-destructive methods to be able to tell the difference in water responses versus, and things like the stomata conductance, closure and opening. So we do this for many plants, and then we look at, their distribution. And here's the example on the right, and you can see that WS stands for water stressed, right? Because they are water stressed, therefore, all those plants have a little bit higher leaf temperature than the WW stands for well watered. And, it looks like there's about, more than 0.5 degree of the difference in their foliar temperature. And this p-value is statistically significant. So one other thing that is quite useful is hyperspectral imaging. So this slide's a little bit cluttered, but on the left here is how we process hyperspectral imaging system. So many of you probably already know that hyperspectral is a data cube. So it's a lot of data, and it's processing a little bit different from you would do with an RGB or thermal infrared camera. So what we do is we extract certain bands out of the data cube, and when do the segmentation, those are the bands that is differentiate the vegetation and the background the most. And then we create a template, and from that template, we can extract all the bands, the information across all the wave bands, and then we would calculate an average spectral reflectance of the leaf pixels from that plants, right? And then we can use this spectral information to develop a machine learning algorithm. And normally that is the partially least squared regression or random forest. And then we can use that to predict the chemical properties of the plants. I put in 4 chemical properties here. So water, nitrogen, phosphorus, and potassium. These are by far the most important nutrients for the plants to grow, and got the economic implications as well if you go to the farm. And so you can see that they are all highly correlated with the spectral data, meaning that you can use the spectral information to predict what's the water content, what is the nitrogen concentration, potassium concentration, or phosphorus concentration. Now I put this publication here, so which is detailed all these experiments and the results. If you are interested, it's published in Frontiers in Plant Science. So if you are interested, that you can look at more about this study. So like Daniel mentioned, right? So we also trying to develop low-cost systems to help with the field phenotyping. And the cytocam is great that you see in the video, but it's also expensive, right? So it is at a fixed location, and you cannot move them easily. So therefore, we wanted to, sort of develop a low-cost portable phenocart system, so that researchers can use it at a broader acreage, right? So this is what we have. So it's quite the concept is quite simple. We got the 2 bicycles, right? We will put them side by side, and then we build a frame to stabilize it. And then we got, as you can see, that there are sensor bars that attach to it, and being able to measure different traits from the plants in the field. And I throw in the map of the Nebraska, and these are essentially the different types of crop that we have tested with this phenocart system. And you can also see the various locations across the Nebraska. We were able to test the system. So by the way, so Lincoln is around here, okay? And then, so you can see that Western Nebraska, we tested with the Camelina, and the distance between these 2 locations is over 400 miles or 640 kilometers. So it's quite, mobile. And if you are interested in, right, so what are the sensors that we use on this phenocart? And this is a close-up view of that, what's going on on that sensor bar. So we have the ultrasonic sensors, right? And then we have, so ultrasonic sensors right here, and that is being used to measure the crop height. And then we also have ocean optics, visible and near infrared spectrometer. And that's the one in the middle. So you don't see the spectrometer. This is the fiber optics, but the fiber optics is leading to, it's coupled to the spectrometer, which is, which is somewhere here on the platform. We got an Rgb camera, which look down right here. And we got an apogee infrared radiometer, and that measures the temperature of the canopy. And finally, which is this one. And then we have the Velodyne LiDAR scanner. So LiDAR stands for light detection and ranging. Basically, it's a laser scanner that return a point clouds of the canopy structure. Those are the hardware of the system, but at the same time, we also need to develop the software, right? And the purpose of the software is quite simple. You have to, synchronize all those sensor systems, and you want to, log the measurement and make sure that the data all you collected all they are stored properly in the proper format on the computer. So we use LabVIEW to develop this software, and you can see that you got some visualization tools on it so that you can actually see the real-time measurement by the system. So you can see the 3D point clouds here of the canopy, and you can see the spectral reflectance. And this here, the 3 images are the, corresponding to 3 plots that you are measuring simultaneously, and that's the overhead images. And there's some other informations there. And I also put the publication associated with this work here. If you are interested, you can look it up. So that's the low-cost system that's we developed. I wanted to throw in this example here because so this is the image that it's a young corn, right, coming from our Phenotipagem system. You have to really know that we developed this system about 6-7 years ago. So that was the time when we don't really, so drone is just start to be widely available. Hasn't been widely available yet back in the time. So if you look at the image, right, so it's really high quality, and you can see a lot of details with the plot that you are measuring. And so the resolution is much higher than what you would get from a drone. And if you do if you look at the images on the right, and that's the results from the image processing. Now, what I wanted to mention to you is that, we can not only separate the corn from the background, right? But we are also be able to distinguish the sunlit fraction of the vegetation versus the shaded fraction of the vegetation. And that is actually quite useful, from various publications mentioned about, if you wanted to estimate the, the efficiency of the photosynthesis or, those kind of things, the physiology side of it, the capability to distinguish the sunlit versus the shaded fraction of the canopy is pretty important. All right, so this is my last slides, and this is another useful case for our Phenotipagem system to score the soybean iron deficiency chlorosis. So soybean here in Nebraska and our northern states like South Dakota and North Dakota, and the iron deficiency chlorosis is really a bigger problem because it's in the alkaline soil, and the iron in the soil is just immobilized. The plants cannot absorb it quite efficiently. So therefore, a lot of the breeding efforts is going in to develop the varieties that are the IDC resistant, right? So, if you are a breeder, you probably know this. So what you do is you go into, you test, hundreds of different genotypes, and you put them, in 2-row or 1-row layout in the field. And then you would send people into the field and manually score them, right? And then the score system that we use here at Nebraska, is one is the most healthy. These are absolutely IDC resistant. Two, you can see that the symptom becomes more and more severe all the way to 9. And this is where really, they are very susceptible to IDC, right? So now the problem with sending the students or the researchers to the field to score the IDC, one is it's very subjective, right? If you ask somebody who doesn't have a whole lot of experience to do this scoring, and you come out, all kinds of results, because the consistency is just not there if you have different people. And then it's really subjective, right? How are you going to tell a 4, versus 5, because they are really close, and it's kind of harder to draw a line between those close classes. So therefore, our when the engineer got a problem like this, right? So we always think, well, maybe we can develop an image processing system to do the IDC classification automatically. And this is what we do with this Phenocart, right? Because we do have a camera on the system. So therefore we take images, right? This is what is shown here. And we do image processing. And then, we sort of, just isolate the plant pixels. And then we analyze the color component of the pixels. One of the fight that we have to fight against is really the weeds, right? So, some of the plots are quite clean, like the one shown here on the top row. Some of the plots are really weedy, right? So, and then we have to do some segmentation technique to remove whatever the weeds in the middle. And you can see, the results here, we are quite successful, but we are not 100% successful. There's always some weeds that's kind of there. It's hard to get rid of by a automated algorithm. But regardless, right? So we do the image processing, and then we look at the color distribution, which I gave 3 examples, right? So you can see that from the red to cyan, that's the color wheel information, and the perfect, the best plants, like the green is the one on the top, right? You can see the distribution is really at the aligned well with the green. And then when you have more severe IDC, the color distribution is gonna move towards the yellow, orange, all the way to brown side of the color scheme, right? And that becomes a really useful information for us to develop a automated system to do the IDC classification. And now, without going through too much details, I just kind of show the results, how we evaluate our machine learning algorithm, right? So RF stands for random forest. And this is, essentially what we have with the automated scoring from 1 to 9. And then, so vertically here, we got the field scores also through 1 to 9. Now, if our machine learning system is perfectly fine, right, perfectly accurate, then you would see all these numbers are aligned at the main diagonal. There will be zeros everywhere else. Now, unfortunately, right, so our machine learning system isn't perfect. Therefore, you see all these non-zero numbers at other places, meaning there is an inconsistency between the machine learning algorithm and what the breeder would score. But luckily, what we see is most of those non-zero numbers are just one class away from the exact match, right? For example, the 6 here is, breeder scores 3 and the machine learning scores 4, right? So, and then, so this is really encouraging because, we certainly see that there is a big advantage, and, it's very promising that we can use this RGB imaging and the machine learning to do the IDC scoring. So this is a 9-class case, right? So as you know, if it is a random, completely random classification, then your overall accuracy is going to be 1 out of 9. So that's roughly 11%. Now, if you look at our accuracy for the random forest, so the accuracy 1 means, those are absolutely 4 on the main diagonal, and that's about 50%. So it's not particularly high, but compared to 11%, that's very good. Now, accuracy 2 is that what we count all those classifications, once one class off as accurate, then our accuracy goes up to 85%, 8%, right? So that was really very useful already for this kind of work. So if you are interested, so I put the publication information there that the field-based soybean IDC scoring using RGB image and statistical learning. So this is really my last slide. So I don't know how we're gonna run this, but I will be here for a little bit more time. And if you have questions, please feel free to leave a question in the chat or ask. Thank you.
So perhaps we can field any questions for Yufeng now. Does anybody have any questions?
Yes, I think that I have a question, 2 questions. I think that the 2 questions is a little bit related, but when you show the LaminaTech greenhouse phenotyping, when you have the trays moving around the greenhouse, and then the plants goes to a chamber where you take pictures and imaging and so and so. How long it takes to run all the plants through the system? And is there any problem with if it takes too long because you have many different influence of light and temperature or daylight or kind of a circadian clock? How do you manage this kind of influence?
Yes, so Paula, thank you very much for that question. And this is a really good question. And right, so that is the limitation, right? So even though we said that this is a high throughput system, but the throughput is still not high enough, right? For many research that we wanted to do. So for our system, if we talk about 8-hour shift, so we can actually finish all 672 parts on the belt, right? So we really have to rush through everything, go through all the imaging chambers. But recognizing that even you start at 8 a.m. and we trying to finish at 4 p.m. You mentioned about the circadian rhythm, and that by the time that those plants get imaging at different time during the day, there will be differences. So that is not so much important for things like if we wanted to measure just the growth, right? And that would be fine. But if we are trying to measure things like very, very fast change in phenotypes, like leaf temperature, then that causes some challenge for us. Yes.
Yeah, I think that is not on leaf temperature, and maybe photosynthesis rate. I think that this is also true for the field imaging, because in the field I think that the influence of the time is a little bit higher, because you can start in the morning and you have difference and to manage. So, and did you, or do you have any kind of maybe of computing or simulation, I think, to avoid this kind of problem? How do you think that this problem could be managed?
Yeah, well, so so for our SpiderCam system, right? So one of the greatest division we have with the SpiderCam system is, we know this is an expensive piece of research infrastructure, right? So it is, you are not going to have a second SpiderCam system or third SpiderCam system everywhere. So what we do is that we use that SpiderCam system really taking high intensity phenotyping activity, right? So for example, we literally run the SpiderCam imaging system. So there's about 130 plots under the footprint. And we scan all those 130 like 6, 7, or 8 times a day, right? Mm-hmm. And what we do is we are looking at the temporal trajectory of the plant responses. right? So, and then we're looking at, mainly just to answer the question, what is the, for example, we talked about the leaf temperature, right? And we know leaf temperature is, it's highly correlated with the soil moisture status, but also the incoming solar radiation and the, vapor pressure deficit, right? But we measure it, multiple times during the day, and being able to figure out really, so what is the time that gives us the best information regarding the crop stress. And then our hope really isn't the spider cam. Our hope is that we can carry that in, we can use that information, and we can develop a low-cost sensor, or a measurement protocol, really, right? And then translate that to the UAV, because we know UAV, you can measure it, at thousands or tens of thousands of acres.
Yeah.
That helps to answer your question.
Yes, for sure. And the case that you showed, using hyperspectral imaging, where you can estimate the concentration of nitrogen, phosphorus, and potassium in leaves. So how does this work? So first you estimate the concentration of the elements by chemical assay, and then you correlate with the spectrum.
Right. That is how we did in that study, and we essentially destructively sampled all the plants, and then we measured the chemical concentration in the leaf, and then we developed the model, right? The signal from hyperspectral imaging and the lab-based measurement, and then we train and validate the model from those two sets of measurement. But once we have the model, so part of it is, the question is, can we really translate this greenhouse-based hyperspectral imaging system to the field, right? And you can see that in our chamber, it is a man-made light. It's not sunlight, and everything is very well controlled. And we do absolute necessary calibration to make that work.
That's right.
It was really difficult, and that you can't control the intensity of the sunlight and probably quality of the sunlight. So therefore, right, so the calibration would be quite challenging, if we have, a drone-based hyperspectral imaging system to look at the crop. So, my colleague Yeyin is gonna touch that a little bit, maybe in her talk.
Yeah. Well, okay, I have another, the last question, I promise. That is,
absolutely.
Well, okay, so thinking in a breeding program where you are selecting for example, for nitrogen use efficiency or things like that. So, and then we are going to use your phenotype system. What are the differences from two genotypes that you can measure accurately measure difference between two genotypes? Thinking about the breeding program.
Yeah, so you're asking about the two different phenotypes?
Yes, exactly. You have two. So in a breeding program, the difference, you are selecting for nitrogen use efficiency, for example. You think that you have a better variety, and then you are going to compare with the other, the wild type, let's say. And then what are the range, the difference that you can accurately measure with your system?
Yeah, well, so our system, so if you're asking about the hyperspectral imaging system, that is really a quantification, right? So we are trying to put everything in a number. It's a quantitative number. So, I understand that a lot of the breeding processes, you don't necessarily need to have an absolute accurate measurement, right? It's all about ranking in the relative performance, one versus the other. So, I think in that case, I think breeding is actually a less strict application compared to what we have, because we truly trying to predict everything to a numerical value. But every method's got its detection limit. It gets an error associated with it. I don't know if you got two genotypes that perform really close to each other, but you got a difference between them. if our system can still tell the difference, that I don't know.
Yeah. Okay. So thank you very much, Yufeng Ge. So I'll ask Daniel again to introduce the another speaker. Thank you very much for a very nice presentation. It was fantastic to see how high throughput phenotypes is progressing and how you are doing so nice in Nebraska. Thank you very much.
Yes, absolutely. Thanks for having me here.
Thanks, Yufeng.
Thank you.
Okay, so the next speaker will be Ning Chai. And do you want to take it away, Yen?
Sure. Let me share my screen really quick.
Yen is in her last year of her PhD thesis.
Sorry. So can you see my screen now?
Yep, I can see it. How about you, Paula?
Yes, fine. Thank you.
Good. So my presentation today will cover mainly the high throughput phenotyping work done in the greenhouse or control environments. So as we all know, plant phenotype is a result from the interactions between genotype and the surrounding environmental factors. Characterizing plant phenotype is very important, especially in plant breeding, to select for the best performing genotype that can adapt to different environments. And with the technology advancement nowadays, high throughput phenotyping platforms that integrate automated data collecting systems and phenotyping sensor has greatly improved the spatial and temporal resolutions of the phenotyping data, which can also allow for large-scale phenotyping data to be collected throughout the entire growth cycle without doing intensive destructive harvest. So phenotyping data can be image-based or non-image-based. One example of the non-image-based phenotyping data is through this spectral radiometer. What it can do is that it measures the reflectance of the leaf, and then different regions of the leaf spectra indicate different information or indicate different physiological information about a leaf. For example, in the visible regions, a typical plant will tend to have stronger absorbance at the blue and red area, whereas there is a higher reflectance at the green area. This is because there is leaf pigments such as chlorophyll that prefer to absorb green and blue and red light and also reflect the green light. This is also why we see plant in green color to our naked eyes. And at the near infrared regions, we typically will see a strong reflectance at this area, and this is because of the presence of spongy mesophyll tissues inside the leaf, which tend to reflect the near infrared light. And at the shortwave infrared regions, we will typically see 3 strong absorption sites. And this is because there's water inside the leaf that will tend to absorb the shortwave infrared light in these regions. And for image-based phenotyping, there are a lot of options, depends on what purpose that you're after. One of the most commonly used sensor is visible red, green, blue camera. It is very robust for monitoring plant growth and plant development. Fluorescent camera, on the other hand, is robust for monitoring chlorophyll fluorescence, senescent area. Infrared camera is useful for detecting the leaf temperatures and also water content, especially when you want to look at the drought stress tolerance in plants. Hyperspectral camera, on the other hand, can it generates hundreds of images of the same plant simultaneously at different wavelengths that spans the visible and infrared regions. So similar to the spectral radiometer, they're both very robust at measuring the stress level and also the physiological activities of a plant. So in my talk today, I will only focus on the use of visible red, green, blue camera and also hyperspectral camera on monitoring plant growth. So here I have a brief workflow of how we use red, green, blue images to monitor plant growth. The idea behind this is that we will need to take several pictures of the plant before we do the destructive harvest. And at the end of the experiment, we'll need to measure plant biomass like dry weight and fresh weight, which will then be used to ground truth the pixel data that we extracted from the plant pictures. To extract the plant pixel from the image, we will first need to segment the area of the plant from the pictures. And this can be done using a green index, which is derived using this formula right here. And in this picture, it shows the plant area that we want to extract the pixel from. And we'll usually do this for 5 side view for each plant. And with the 5 side view, we'll then take the average and then correlate with the fresh weight or dry weight to get this model. And with this model, we can then kind of estimate the biomass from the pictures that we took before the harvest and generate a growth curve to study the growth, the biomass accumulation pattern for the plant. And RGB images, like Yufeng say, not only can monitor plant growth, it is very useful also to monitor plant height developments, leaf area, and disease progressions over time. Here I have another workflow of extracting leaf spectra data from hyperspectral images. And like I mentioned earlier, hyperspectral camera can simultaneously generate hundreds of images of the same plant at different wavelengths that spans the visible and infrared regions. So the first thing we need to do is to separate the plant regions from the background. And this is usually done using near normalized different vegetation index, which is derived using the wavelength, using the reflectance at the near infrared and the red regions. This NDVI is also widely used to assess plant health status. For example, a healthier plant will typically have higher reflectance at the near infrared regions and also strong absorbance at the red area. So when compared to a stressed plant, the NDVI of a healthier plant will normally be higher than the stressed plant. So back to the data analysis. In this case, we use the image at the wavelength 670 nanometer as the red and the wavelength at 770 nanometer as the near infrared to compute the NDVI. And you can see that the plant regions look significantly brighter than the background. And this will allow us to easily separate the plant regions from the background. So if you're interested in just the leaf area, there's also another trick where we can separate the leaf from the stem. This can be done by selecting a wavelength at which the leaf reflectance is different from the stem reflectance. In this case, we use the wavelength at 1160 nanometer. You can see that the leaf area is significantly brighter than the stem area. And this can allow us to kind of just segment the leaf and excluding the stem and the background. And once we define the area of the leaf, we can then easily derive the leaf reflectance curve from the hyperspectral images. Here I have another workflow that describes how we can predict leaf biochemical data from leaf reflectance. So like I mentioned earlier, leaf reflectance not only can give us an idea of the leaf pigments and water content of a leaf, it can also be used to give us some important information about biochemical properties. To do this, one option to go is to use partial least square modeling, which will correlate the leaf reflectance data with the ground truth leaf nitrogen data. So this plot right here shows the relative importance of each wavelength at predicting leaf nitrogen content. And as you can see, there are only a few regions that are important for predicting leaf nitrogen, where there are a large amount of wavelengths that are kind of redundant and not useful for predicting leaf nitrogen. So what partial least square modeling does here is that it can reduce the dimension of the input spectral data and assign loading with different magnitudes to each wavelength depending or based on the importance of each wavelength at predicting leaf nitrogen. So here we kind of plot the ground truth nitrogen data against the predicted nitrogen data that we obtain using this partial least square model. And you can see we got a pretty good R-square value. And then with this model, we can then easily estimate the nitrogen accumulation over time with the reflectance data that we measure beforehand. So here we have black curve representing the plant that receive high nitrogen treatments and red curve representing the plant that are subjected to drought to nitrogen stress. We can see that the nitrogen plant have lower leaf nitrogen level as compared to the high nitrogen plant. And starting from day 55 onward, there is an increment in the leaf nitrogen content. And this was because we added some more nitrogen fertilizer at this time point. So we can see that this hyperspectral images was pretty robust at detecting the change in leaf nitrogen level. So from here onwards, I have two experiments to show you. The first one is demonstrating the use of high throughput phenotyping in studying plant microbe interactions. So this project was done in the high throughput phenotyping greenhouse that Daniel and Yufeng show. The objective of this study was to determine sorghum growth response to microbial inoculations under low nitrogen conditions. And the rationale behind this is that we think some of the microbes that were originally isolated from sorghum may be beneficial for plant growth under stress conditions. So what we did was that we inoculate sorghum with synthetic bacterial community under both high and low nitrogen conditions. And to monitor the plant growth, we used the RGB images and took picture of the plant every other day. We also tracked the nitrogen accumulation over time using Hyperspectral Camera. And the phenotyping was done for 2 months. And at the end of the experiment, we measure sorghum fresh and dry weight, and also we measure the leaf nitrogen content from the most recently fully expanded leaf as our ground truth data. So these two pictures right here shows you the development of plant under high and low nitrogen conditions. And then this picture here shows you how the plant look under the hyperspectral camera. So we have some result right here. In the plot on the left shows you the biomass developments of this genotype grasses, starting from day 25 to day 65. And this data was derived from the RGB images that we took. We use different colors to represent different microbial cocktail. For example, we use blue to represent uninoculated control, and brown represent cocktail 1, gray represent cocktail 2. Cocktail 1 and 2 are consisting of the bacteria that we isolated from sorghum rhizosphere and root endosphere. Cocktail 3 represent the bacteria isolated from soil colonizers. Cocktail 4 is just a combination of C1 to C3. And cocktail 5 is mainly the root colonizer that we isolated from sorghum. So as a result, we can see that under low nitrogen conditions, inoculating this sorghum genotype with all microbial cocktail seems to have a suppressing effect on their growth as compared to the uninoculated control. And similarly, we see similar trends on the leaf nitrogen content. This leaf nitrogen content accumulation curve was obtained using hyperspectral images. And we can see that compared to the uninoculated control, all the inoculated plants seems to have lower leaf nitrogen content. And the increments here starting from day 54 was because we added some nitrogen fertilizer at this time point. So in addition to the use of expensive high throughput phenotyping platform, here we have another experiment that used a low cost DIY platform to monitor soybean growth under alkaline conditions. This low-cost DIY platform is developed by Dr. Yufeng Ge, and it costs about $8,000, and it's pretty simple. It only consists of a rotation platform, which shows in all three pictures in this slide. And this rotation platform will rotate the plant as you take picture, and this rotation platform is connected to this motion controller system, which is this little white square box. What it does is that it serves as a connector between the rotation platform and our computer. So we can control the rotation platform by communicating with this motion controller system. And of course, another thing that we need is a camera and a tripod. You don't need to have a super good quality or expensive camera to do this. All we used was a cheap Logitech webcam camera. And then when you do the phenotyping, we will also need a black background. And the computer program that we use to do the phenotyping is called LabVIEW 2017. And the program that we use for downstream image processing is MATLAB. So here we have two pictures of soybean under neutral and alkaline conditions. You can see under alkaline conditions, the soybean will grow kind of yellow because of the iron deficient chlorosis. Here I have some results from that experiment. So before I go into the results, I will provide you with a little background. So about 30% of the land on Earth is alkaline. Alkaline soil can cause iron deficient chlorosis in soybean. Approximately $260 million are lost annually in the north central regions of US due to soybean iron deficient chlorosis. So the objective of this experiment was to determine soybean growth respond to microbial inoculations under alkaline conditions. And in this case, we use a pH of 8. For the phenotyping, for each plant, we took a total of 8 site views to make sure that we get the most accurate phenotyping data. And for the ground truthing, we measure soybean fresh and dry weight at the end of this experiment. And for the microbial cocktail, we have, of course, an uninoculated control, which is represented by green, and we have cocktail 1 and 2, which are the bacteria that we isolated from the alkaline soil, and cocktail 3 and 4 are the bacteria that we isolated from neutral soil. So as a result, under neutral conditions, we can see that the inoculations of cocktail 1, 2, and 3 seems to have a growth promoting effect on soybean, whereas cocktail 4 doesn't seem to have any growth promoting effect because it looked just similar. The growth curve of plant inoculated with C4 just looks similar to the uninoculated control. And under alkaline conditions, none of the microbial cocktail that we inoculate have any growth promoting effect on soybean. So in conclusion, we shows that it is possible to get good phenotyping data using cheap DIY phenotyping system. So you don't necessarily need to use a super expensive high throughput phenotyping platform. And with that, I will thank you for listening. And if you have any questions.
So thank you very much. Ning Chai, thank you for the nice presentation and for the data that you collected looking for the role of microbes and the response to nutrient acquisition. And this is really very, very, very important. And I am a little bit surprised that in the nitrogen, low nitrogen concentration, you have a negative impact of the microbial inoculation. And this is a little bit surprised because it is expected that beneficial microbes can help plants by fixing nitrogen from the air. So is there nitrogen fixation capacity in the microbial community that you inoculated the sorghum plants under low nitrogen condition?
The answer is no, because we tested the bacteria that we use for nitrogen fixation abilities using acetylene reduction assay, and it seems like only 1 or 2 have positive response to the acetylene reduction assay. So I would assume that the bacterial community that we use to inoculate sorghum did not have much of a nitrogen fixation abilities.
Yeah, because it is expected that at least here in Brazil, there is a notion that there are some nitrogen fixing bacteria that is associated with grasses like sugarcane, corn, or sorghum that helps plant to fix the nitrogen and in under low nitrogen concentration. Maybe this suppression that you saw, maybe, I don't know if Daniel could help with this question also, but maybe with plants, the inoculate bacteria can maybe change, the physiological status of the plant so that it cannot take up nitrogen at low concentration in the soil.
Yeah, I mean, I think what we tested, Yen tested multiple genotypes. So you didn't see that response for all the genotypes, right?
No, we detected a strong genotypic response. So this genotype shows like strong growth suppressions after we inoculate the bacterial community. But for other genotype, most of them didn't respond, or the growth did not change with bacterial inoculations. So it's also depending on the genotype that you inoculate.
Yeah.
Yeah, so this is a genotype that we find is fairly sensitive in the field to low nitrogen. And so, you can speculate based on Yen's results that maybe there is some sort of competition between the plants and the microbes under
Yeah.
these conditions of very low nitrogen. And this plant genotype, it just can't compete, basically, or it doesn't compete as well as some of the other ones. So originally we were planning to test nitrogen fixing microbes, but there are various on our road to testing and our planning, we were derailed by some preliminary early results that we couldn't then repeat.
Yeah. So that's very interesting also that you were able to quantify the nitrogen using hyperspectral imaging, and it was very nice. So one question that I would like to ask also maybe for Daniel is that, this LambdaTech greenhouse, is this, what kind of because I have heard that this, I think that you told me that, well, this is a quite expensive, let's say, facility, and it's kind of the cost per plant that you put in an experiment. Could you tell a little bit more about this, Daniel, maybe?
Yeah, this is one of the reasons why we wanted to develop like this cheaper version that Yeyin showed, where we can just do it in our own greenhouses. But right now it's like $3 per plant per day. So let's say you have 600 plants, that's $200 a day, or about $1,500 a week for an experiment. So, if you want to grow plants for 3 months, that really adds up. And so experiments could be in the order of $30,000 US. And you just can't do that many of these large experiments because of the cost. We have really great people who manage the whole facility, so that's nice. That is part of the cost, but we would love other people to come and use the facility.
Yeah, good. Fantastic. So thank you very much, Yeyin, for your kind of nice presentation. And I think that we can move now, Daniel, to the last speaker.
Okay, so the last speaker is Yeyin Shi. You want to take it away, Yeyin?
Sure, of course. Let me try to. So if I share the slides, and then if I go to the presentation mode, can you see the slides in the presentation mode?
Yes. Yeah. It's
okay,
fine.
Awesome. And first, like to thank you, Dr. Arruda and Dr. Schachtman for the invitation to this open class. And also thank you for Paula and then Daniel's help so far. So Ola and then Boa Noiti. So if I'm doing it in a right way. So my name is Yeyin Shi. I'm an assistant professor in biological systems engineering at University of Nebraska-Lincoln. So my research is intensively focused on drone technologies for high throughput plant phenotyping and also precision and then digital agriculture applications. So today, I like to talk 3 main topics here. The first one is the drone types and then flight operations, and then talking about the sensors that can be used on the drones, And then I know many of you are interested in the data processing analysis part, so that's going to be one of the main topics there. Okay, so first, some terminologies here. We use drone all the time. It's pretty much like a informal but widely accepted term. And also you might heard about terminologies like UAV or UAS. So UAV stands for unmanned aerial vehicles, and it's also commonly used. UAS really stands for unmanned aircraft systems. So that's the definition by United States and Federal Aviation Administration. And so the reason why it initially being proposed there is that the Federal Aviation Administration really wants to refer to a system rather than just a drone itself. And then some of you may heard about this new term here, that unoccupied aircraft system, which the acronym is also UAS. That's really because of people wants to say that with the current regulations, there still needs to be pilots on site. So it's not really unmanned, it's just unoccupied in the vehicles. So before we talk about the drone, I'd like to mention about the different remote sensing platforms that are commonly used in agriculture. We have satellite, we have airplanes, and then also nowadays we have the drones. I always like to show this figure that I found online about the relative locations of each of those platforms where they are operating there. So satellites, they're really far away from us. They're about 700 to 10,000 kilometers away from us. They're just orbiting in those close to, Earth, those orbits. And then the aircraft and then drones, if they are doing something like the sensing, they really operate in the most inside this atmosphere layers around the Earth called troposphere. So aircrafts, they operate usually in about 3,000 feet, or about, I believe it's about 10,000 kilometers, 1,000 meters from us, sorry. And then the drones are, with the current regulations, they could only fly up to 400 feet or 120 meters above the ground level. So all those are actually great data generation sources for agriculture. And I do see some of the breeding studies, they also use the satellite imagery to collect the data. And of course, those are high spatial resolution satellite imagery with the spatial resolution maybe around a meter or sub-meter level. And the good thing about satellite imagery is that you can really have it at anywhere. And then you don't really need to have, like, say, a pilot to actually go on site to collect the data. But then still for breeding, for field-based breeding or phenotyping, I think drones serves the most roles, is because they provide the most high spatial resolution data. Different types of drones, you see those we call fixed wing drones. They really like a miniature of the regular airplanes. And so their wings are so-called fixed. They can't move. The good thing about them is that they're really power efficient. So as long as they keep a certain forward speed, the shape of the wings will generate a uplifting force themselves. So they can just slide without even using the power. But then the other side is they can hover, and then they have to keep a certain speed. And most of them do need a runway to either to take off or landing. So in the phenotyping area, most of the studies are using this so-called Rotary Wingstrongs, which are the ones with multiple rotors there. So the good thing about them is that they can hover, and then they are really nimble and easily to be controlled during the operation. And also they are, they're relatively stable. So the data quality is good. There is another type of drone that is called hybrid, which is a combination of the fixed wing and then the rotary wing. But then the technologies there are not really that mature. So it hasn't been very much adopted yet. And I'm not sure about the regulation in Brazil, but then here in the United States, the regulations for drones is a big thing. First is that every time we have a drone that is more than 0.55 pounds or 250 grams, that will have to register this drone with the Federal Aviation Administration. And then not everyone can fly the drone for the commercial purposes. And also doing the research is considered as a commercial purpose use. If we want to do this, then we have to have a so-called Part 107 remote pilot certificate. And this one, even with this certificate, we have to fly less than 400 feet, and then within the line of sight. And really, we can't fly in those restricted airspace, even though there are ways to bypass them. So like say the studies that Daniel and we conducted all the way to Scottsbluff in Nebraska, that one we actually applied for the special approval for the flights. And if we really want to go beyond those what is approved by Part 107 certificate, then the next one that we need to apply for is called this COA, or Certificate of Waiver or Authorization. So I just like to mention this because I'm not sure the regulation in Brazil, but then this is just a important thing that every drone pilots, or if you want to do a study with a drone, that you have to consider this one. So nowadays the drone flights is very simple, very easy. Everyone, if you follow the manual carefully, and then carefully do some test flight, you'll be able to fly a drone. So the system consists like a drone itself, and then what you have on the ground or with you in your hand is a remote controller. So it's basically similar as those RF links, those remote controller that we play when we are small as a toy. And then you can hook a smartphone or a tablet to that remote controller, and it serves as a flight planning and also a visualization purpose. So if we want to use the drones for research, and especially for phenotyping, then we really need to do something like planning for the path for the flight. So we can pre-plan for a regular path that is covering the whole area of interest. And then this one will be based on, for example, what type of camera, what's the field of view of camera, and then what the flight height. And also another important thing is we need to consider the overlaps in between the images, and which we're going to talk about later in this lecture. So here I'm just showing a couple of videos about this drone takeoff. Okay, how can I? So this is a takeoff of a fixed wing system. And then for a rotary wing system, this is what we call vertical takeoff and landing. Okay, so next part, let's talk about the sensors that can be used on the drone. And the first category that the previous presenters has already been talked about is the RGB camera, or sometimes people will also call it natural color camera, because they simulated our human eyes. And so the output is very similar to what our human eyes see. So it has RGB, red, green, blue, the 3 bands, and most of the cameras that come along with the drones already have this. Is already this RGB cameras. So here I'd like to briefly mention about the electromagnetic spectrum. So all the lights that is around us, or the energy around us, are basically falls into this electromagnetic spectrum. So from the left to right, the wavelengths increase, and then the frequencies decrease. So what our human eyes can see are really falls into this visible range, which is about 0.4 to 0.75 nanometers. However, in agriculture, we often will also use this near infrared, infrared region. So this infrared region can be further divided into the near infrared, mid infrared, and then the thermal infrared. So the reason why our human eyes see the plants are, most of them in the green color, is because most of the plants, they make use of the red and then the blue regions of the electromagnetic wavelengths for photosynthesis. So the green ones are actually not being much used, so they reflect it out. And also, if we're talking about the near infrared region, some of them will be reflected back, and some of them will just transmit it. So the near infrared one are kind of translucent, or the leaves are pretty much translucent for the near infrared lights. And if we're looking at the canopy level, so the canopy level, all the, like say the red and then the blue lights are being largely absorbed and utilized by the first layer of the canopy, and then the green lights will actually reflect it back. And then the near infrared one they just transmitted through the canopy layers and all the way to the ground, and then they will bounce back. And so because of this principles, we can make use of this different reflectance from the RGB and then the near infrared spectral regions to estimate how the canopy structure and how the leaf, how the inside structure is. So a sensor or a camera that really make use of this principle is called multi-spectral camera. So it's usually have about 3 to 10 spectral bands. And then there is usually 1 or 2 spectral bands there are the near infrared bands. So you might heard about those vegetation indices, for example, normalized difference vegetation indices, NDVI, or normalized difference red edge index, the NDRE. They are basically a very simple mathematical equations that is calculating the difference or the normalized difference in between the near infrared and the red, or the near infrared and then the red edge band. And one of the very popular multispectral camera on the market, the MakerSense camera, multispectral camera has 1, 2, 3, 4, and 5 spectral bands in those RGB, red edge, and the near infrared region. So with these camera, we can better estimate the plants, for example, their health, their stress level, or their nitrogen or their chlorophyll contents. Okay, so we talked about the multispectral camera. Next one that we're going to talk about is the hyperspectral imager. So this is a hyperspectral imager that we got from hat wall system. So this hat wall hyperspectral imager, yeah, here is the zooming picture of it. So instead of have just like 5 spectral bands there, it actually has 270 spectral bands, but they are narrow, very narrow spectral bands. So if we plot them out, actually you can almost see a continuous curve here, but it's actually still discrete. It's just when we plot them out, we can see the continuous profile. So with this kind of cameras, well, I actually don't want to call it a camera because it's actually that's the line scanning, and then in order to form like a 3D view, we need to, I mean, the imager needs to be moved so that to form a 3D view. So this is a study that we collected over a turf grass field, that it has treatments with irrigation and also treatments with different nitrogen levels. So we can see that this, for example, this yellow dash line here is this well watered, and then nitrogen level 6, which is the most of the nitrogen applied there. So they have a high reflectance in the near infrared region, and then a relatively lower ones in the visible region. And this one, this pink and then this blue one are actually this water stressed and then nitrogen level 1, and then water stressed nitrogen level 3. So they have lower or much lower reflectance in the near infrared region, but then relatively higher reflectance in the visible region. So So was this hyperspectral sensor similar as what Yan and then Dr. Ge mentioned earlier? We really have more information here that we can investigate in to see the different properties of the plants. And then also another type of cameras is this thermal camera. So thermal camera, as we just talked about, it's a long wave infrared. It detects long wave infrared radiation. So basically, it measures the temperature. So higher the temperature, then stronger the radiation intensity. So that's the reason why thermal cameras has been widely used to detect the water stress or any of the irrigation issues in the field. Basically, anything that can cause a reduced evapotranspiration and then further cause a change of the canopy temperature that could be possibly detected by the thermal camera. However, there is an issue using the drone-based thermal camera, is that the thermal, as we just talked about, the thermal camera is really detecting or measuring the temperature of the canopy. However, if there's any environment influencing factors there, for example, if there is wind or if there's any change of the solar radiation, this change of the environment could actually cause larger temperature change than the actual temperature change caused by, say, the water availability or the health of the plants. So in that case, if we take an image here at part of the field, and then we take another image at another part, sometimes it's hard for them to be compared. So that's something that we need to just keep in mind when we use this thermal camera, these technologies. And then we also mentioned about the LiDAR, light detection and ranging technology. So it really just used the time of flight principles to measure the distance in between the light source and then the target. And then if we have a little motor there to rotate the light source, then we can actually form a line of scanning. And then if the platform is moving too, then we can form a 3D scanning for the object. So LiDAR technologies has been used a lot for, say, canopy height measurement or canopy structure profile. And then actually, the image I just showed you, right by the hyperspectral imager, we have this LiDAR sensor there. And then if we can really collect good quality of data, then the LiDAR sensor can actually provide us more accurate and more precise canopy structure profile than the digital surface model generated by the image stitching and also the structure from motion algorithm. However, a problem that we need to consider here is the cost of the system and also the cost of the data processing and computation. So it's much more complicated to process the LiDAR point cloud data rather than a digital surface model, like say the 2D image. Okay, so the last part is the data processing, analysis, and applications in high throughput plant phenotyping. First thing is, we've already mentioned that since the drone flies relatively close to the Earth's surface, each of those images actually only cover a small footprint. So, but then in phenotyping, we want a map of the field. So the first step is to stitch all the images to a map. And then thanks for some of the commercial software there, like Pix4D or Agisoft. They've been developed over the years, and they've give us very good results there on the stitching. So the output of those stitching software are really two major things. One is we call orthomosaic, which is the map. And then another one is the digital surface model, which is basically the elevation model of the field. And then we could further derive the vegetation index maps using the orthomosaic. For example, if we use a 5-band multi-spectral camera, then we can calculate various vegetation indices. For example, NDVI, NDRE, GNDVI, whichever ones that is suitable for your study. And we can do this throughout the season so that we can have this time series data or the maps. And then we could also have the plant height map. So this one we really just needs to use the same, to calibrate all the maps with the same ground level so that we can see the progress of the canopy throughout the season. Actually, we can derive much more information than just vegetation indices. And then there is actually a big advantage of doing that. One of the studies going on right now for us is to estimate the leaf area index from the drone's data. So the leaf area index is, I believe many of you are familiar with it, is an index that is highly correlated with the, say, the biomass accumulation. And also a lot of the crop growth models are actually use the leaf area index as an important parameters to calibrate the model. This is a study that we did over a hybrid wheat field with 40 genotypes in 2019 and then 2020 season. We collected those, we called ground truth LAI using a handheld LAI meter. And then we collected those drone imagery at 4 dates throughout the season. 2 of them are before flowering and 2 of them are after the flowering stage. So if we calculate the NDVI from the drone's imagery, and then if we plotted them versus this handheld sensor measure, the LAI, we really found not very strong correlations there. If we're talking about all the data sets, even though there are sort of some correlations there for each of those groups or the dates of the data we collected. And then if we look at this NDRE, this vegetation index, there is really not much strong correlation there. It's just a cluster of data points. G and DVI, some of them there for some of the dates, but then if we look at the overall data sets in total, still not. The correlation is only about 0.16. Canopy cover, still not much good correlation. And then plant height, not much there. So this really makes us to investigate more is how can we really make use of the data to accurately estimate the leaf area index. So instead of looking at the individual variables correlation with the leaf area index, we extracted 3 main features or the traits from the drone-based data. We extracted the vegetation indices, which are those vegetation indices that I just showed you, the canopy cover based on the image processing, and then the plant height measured by the drones. And then we feed them into an artificial neural network. And then the output is the LAI. And then we use part of the data we collected for training and then part of them for testing. And then surprisingly, with this method, we have a relatively high or strong correlations there in between the model estimated LAI with the manually measured LAI. So this really tells us that if you don't see good results with some of the information or some of the traits you extracted, maybe try some others, and then the results may be improved. So there are many other applications for the drones data. This is a study that Daniel and I we did a couple of years ago, and then it's on estimating the biomass for 24 genotypes of sorghum under the different nitrogen treatments. So we also investigated multiple traits extracted from the data. For example, plant height, ground cover, a couple of classical vegetation indices. And then we found that the correlation there were some correlations or strong correlations with the fresh biomass and also the dry biomass there. And then we also look at the correlations in between the drone estimated chlorophyll and the nitrogen contents with the chlorophyll and the nitrogen contents that we did the samples with. There are also some strong correlations there. And then, so Yeyin just talked about the ASD hyperspectral system. So if we use the similar spectral regions in the ASD hyperspectral sensor, we actually got very similar performance with the drone collected data over the field. So that's pretty encouraging. Another thing that we actually just recently finished was also with Daniel's group is about the drought tolerance evaluation using the drone-based thermal and then multispectral data. So we have those well watered and then those water stressed plots. And then so our goal is to develop a way to evaluate the drought tolerance So the metrics, one of the metrics that we looking at here is this geometric mean productivity, which is like say the square roots of this biomass, the well-watered biomass multiplied by the water-stressed biomass. So with this, we actually found that if we use this multiple of data source together, we actually have the strongest or the highest correlations there for the GMP, this metrics there. So this is actually a encouraging results, because if you think about evaluate all those drought tolerance in the field, that involves a lot of, like say, biomass harvesting and then a lot of ground truthing there. This is not an easy task. So if we could have the drone system to help with this process, then it's quite encouraging. And then some of the previous presenters already talked about the machine learning, deep learning based analysis. And then this is whenever that we found a regular, say, statistical analysis, or say simple linear regression or multiple linear regression. If they could not serve well, then we'll always go to those machine deep learning type of models. We can use them to detect and locate the tassel of the maize, or we can use them to automatically delineate the lodging area of the winter wheat. Or as Dr. Ge mentioned earlier about this soybean iron deficiency chlorosis, just a rating, a automatic rating for the system. Or we can detect and locate the weeds in the fields. Okay, so last, I'd like to mention a couple of emerging technologies for drone that can be possibly used in agriculture. First thing is, there is recently commercialized drone-based RTK GPS system. So for those of you who are not very familiar with this technology, is that the regular GPS on board of a drone system is the differential GPS system, and they usually have a positioning accuracy at about, say, 3 meters. So you can think about every time the drone, the position that is logged by the image, the accuracy of the position will be 3 meters accurate. But then the current technologies can have this RTK GPS on board, which is really provide us about the centimeter level accuracy. So if we will have a drone system that we launch at a point, most likely if we click on this return to home, the button there, then we can have this system return to its original point that it launched from. And also, it can also have a great application in the phenotyping application, is that we can use that one to have a more accurate canopy structure profile, and then we can precisely overlay the time series data over each other. And another technologies is to using the drones for spraying. So this is something that is getting started in the United States, especially for those areas that like the big aircraft or the ground spraying systems that cannot easily accessible, then this drone-based spraying system really plays a role there. So we have people that is working on, for example, like the noxious weeds, those invasive weeds along the waterways, having used this commercial system to manage the weeds. And also by talking to Dr. Ge earlier, is that this one is possible it can be used for phenotyping and then to spray some of the chemicals to the plots. Okay, so that's it, all I have. And then I think we probably run out of time here, but then thank you very much for your attention.
Okay, so thank you very much, Yeyin Shi, for your let's say a wide view of use of drones for many different applications and generation of data. And when I was thinking that yes, it's you generate big amount of data, and I think that there are ways of managing, automatic processing of data, but not necessarily the automation of data processing are capable of managing, let's say, variations that you can have in your image. All right. For example, if you are taking image and you have a little bit of cloud because the changes in the weather, and the question here is, with this big amount of data, how easy is for, let's say, for a researcher or PhD students or postdocs, to extract all the important information you collect from all those data. So how easy is to manage the data?
Yeah, so that's actually a very good question, Paulo. I think first thing is we really need to have a good understanding about which sensor to choose or which data to collect. So based on your interest, right? So if you're more interested in the canopy structure or the biomass accumulation, or you're more interested in, say, the stress, or is it a nitrogen stress, or is it a, say, water stress there? So first is to determine what would be the most promising data to collect. So that will narrow down the data collections there. And then the second part is once we have those data sets, it's really what are the informations that we really need to extract from the data. So there are probably two different ways for this. Sometimes people think about, oh, let's just extract those more explicit or more specific thing. For example, a certain vegetation indices, or say the plant height or the canopy cover, because we know those will be correlated or strongly correlated with what we're interested in. And there's another opinion, it's just let's just throw in all the data and then extract whichever that we can extract. And then you probably heard about some people talking about the latent phenotypes. so those to me are really the phenotypes that we cannot explicitly describe. But then just let's say a machine deep learning algorithm to extract hundreds or thousands of features out, and then just let the models to decide which one to use. But then actually people would always think about, like, say, okay, so outdoor, like say drone-based phenotyping, you have lots of data. It is actually probably not as much as, say, an indoor greenhouse-based or like say ground-based platform, because you would only have, for example, we have great students here that is working very hard all the time. But then during the hot summer here, or the, like say the winter with the chilly winter here, we could only collect a certain amount of data. And then most of the phenotyping field, they are not as a, for example, like the quarter section, the irrigated large area. Usually they are, say, 3 acres, 4 acres, or like maybe like 10, 12 acres large. So it's relatively smaller field. And then so in terms of the actual data volume side, it's actually not that much. Rather than, I mean, compare with if you have a greenhouse system that is continuously collecting the imagery all the time. Continuously.
It is something that graduate students can definitely do. Like Ning Chai, she works in my lab, and she's been trained by someone in Yufeng's lab, and she can now do all this stuff herself. And then for Yeyin, her work is largely done by students. You know, it helps if they have some sort of mathematical, or they want to work with computers. Yeah, so it's definitely doable, and there are programs out there. And I know, like for the MicaSense camera that Yeyin mentioned, you can upload images to the company website, and that'll help you process it.
Yeah. So there are some few questions here that I think that we have already discussed it.
So I think that can be used to measure. Sorry.
Yes. Sorry. Go ahead.
And would you like to finish up? I know we're over time.
Yeah, a little bit, but one question is from Paulo Herman from Embrapa Instrumentation. Embrapa is they have a station or a unit of Embrapa that is working hard in the instrumentation, including drones and everything. And then the question is overall, what is the gain in terms of what have been done over the last maybe many years for breeders, and what those new technologies of high throughput phenotyping is really bringing into the table for the breeders or even for the farmers. So
can I take a shot first? I'll then go over to Yeyin. So, you know, there's a number of gains. One, it's much easier to study the dynamics of what's happening with plants over time. So to do some of the experiments that Ning Chai showed, we would have to have 10 times as many plants, and we would have to be constantly doing destructive harvest. So in terms of dynamics, these tools are fantastic. In terms of large field experiments, we can cut down the costs as well by using some of these imaging methods. We've been doing stuff with nitrogen use efficiency, mapping, nitrogen levels in leaves and chlorophyll. And we can do that in 2 days, and we only have to take about 100 samples for the ground truthing. So we save a lot of lab time and money there. and then in terms of breeding, precision farming, I think a lot of people here want to use drones or computer vision to find diseases. So it'll save a lot of time in people walking the fields looking for diseases. I don't know how much progress is there. There's some applications to nitrogen applications. So one of the professors here developed a just green seeker sensor that sits out on booms on a tractor and would dynamically tell the tractor how much nitrogen to add. So it hasn't been widely adapted, but it is something that's out there. And how about you, Yeyin?
Yeah.
Yeah, I really agree with what you just said. Daniel, is I think, I mean, I'm an engineer, and then I think eventually it needs to be, I mean, the plant scientist to answer this question. But then from a engineer's point of view, I think what we're trying to do is provide the automatic system to facilitate the whole process. And then another thing is not only, I mean, by saving the labor and then the time, but also we're providing the documented way, right? So if you only, for example, if you only making like manually making the notes about, say, plant height or the rating there, for example, the disease or the tolerance level, then there are some subjective errors there. Or if you really wanted to going back and then to the historical data, see what actually going on there. So we will have the maps there, we'll have the imagery there that we can easily goes back and then to check. So but then yeah, I think that question is actually maybe a very common question there, is especially in terms of right now, like the maturity level of the system. And
so
then
what are the
the height.
challenges? What do you
Okay.
think are the challenges for engineers?
Okay.
Yeah, I think the one of the most challenges there is that we are talking about the indirect measurement there, right? So we are, instead of actually using a ruler or using something to measure, we are using something, say the image, the cameras, or yeah, so those are all indirect measurement ways. So there are limitations there.
Yeah, that's great. Yeah, I think we probably.
Yeah.
You want to wrap up, Paulo?
Yes, okay, so thank you very much, Daniel, Yeyin Shi, and Ning Chai, and Yufeng Ge, for the nice presentation and a nice overview of using different approach for high throughput phenotyping. I think it was great, fantastic, and I'm sure that there will be many questions that the audience is gonna send to us, and we will send to you. And thank you very much, and thank you for all the attendees that are here with us. And next week we are gonna have a talk from Juliana from our team that's gonna talk about experimental design, how to do experimental design to extract the best information from field trials and or from greenhouse experiments. And this is going to be done by Juliana next week. So thank you very much for all of you. Thank you, Daniel, again. And thank you for all the nice information that you brought for us today. Thank you. Have a nice end of the day. Thank you.
Take care. Bye-bye.
Take care. Take
Thank
care,
you.
you all. Take care. Bye-bye.